Everybody for coming, and we're going to have the last talk given by Joe Gerarci, who is the Chief Scientific Officer and the Chief Technical Officer and the co-founder of Netramark.
Hi, everybody. Can you guys hear me okay? Yeah, I'm coming through. Okay, so I thought it would be appropriate with this group to talk a little bit about Netramark, what we do, but also to talk to you about my journey so that you guys can perhaps feel like there is a path ahead of you that you may have not considered or that you're desperately trying to consider. So let me tell you a bit about my story. So I was doing my math undergrad here along with neuroscience and some biochemistry. But I became obsessed with quantum gravity and nonlinear PDEs and all that stuff. So I just stuck to the math, ended up getting a bachelor's here. I got a master's as well in mathematics. And then I started a PhD in pure math, but a physicist that was here gave me a problem in quantum computation. And that changed my life because I put a dent into this problem that was deemed important by DARPA in the United States, their research military branch. And so they invited me to go move to the beach in Los Angeles and do math and play basketball, and that's what I did. So I went, and what I did is I organized it so that, having already passed all my comprehensives, I made it possible so that I was shared between University of Toronto and University of Southern California. And then I successfully figured out some cool stuff about quantum algorithms and the limits of quantum computation. And I finished that PhD and was offered a job at Harvard, at the Perimeter Institute, and some Quantum Institute in Calgary, and some cancer stuff, right? So, right, it was the beginning of this, what can we do with AI? And most people weren't talking about machine learning yet. And so I went to Princess Margaret Hospital. I came back from LA and I joined Princess Margaret Hospital as a postdoc under an award I won from the Canadian government. And I started learning about cancer as a mathematician. And I was thrilled to learn that cancer is essentially a graph theory problem, a very, very, very complex graph theory problem. And shortly after that, I won an award from NSERC that came with money. And it's the kind of money that you have to take to a company, to a startup. So I said, you know what, I'm going to take this shot and I'm going to go join this company while maintaining my academic relationships. So I continued doing a postdoc and I was invited to be faculty at Queen's University. So I became an associate professor and I was associated with the University of Toronto and also with this startup working in cancer biomarkers. And the NSERC covered a postdoc basically where I was doing AI development way back. Like I won this award in 2009. And so this was an amazing launching pad for me to start to understand what are the critical problems when it comes to understanding cancer and neurodegeneration. So I did a bunch of work and I was thrilled to find out that neurological issues are just really, really complex graph theory problems. And so I started creating a unified mathematical approach to understanding the network problems that define oncology, the network problems that define brain anomalies, whether it's depression or schizophrenia or Alzheimer's disease. And that led to an award from Eli Lilly, a large pharmaceutical company, which then put me in the faculty of medicine in University of Toronto, and I started working at Toronto General Hospital, Toronto Western Hospital, and eventually CAMH. And I started becoming the data guy, started becoming the mathematician who create these weird models and also build these machine learning algorithms. And then what happened was I had an opportunity to become a full-time academic, continue going down the tenure path position, but I discovered something that at the core is what I'm going to show you a bit about today. I mean, it's grown immensely since then, but I had this very difficult choice. I dedicated my life to becoming a mathematician and a medical scientist, and I had to figure out if I wanted to give the entrepreneurial path a shot. So what I did is I went for it. I quit all my jobs, except for my association at Queen's, because I was still publishing papers. And I went to Equifax, you know, the people that do yeah, right. And I took over the machine learning department, and I hung out in Atlanta, and I hung out in Toronto, and I did that, and I grew practice there where I was able to use all of your records and experiment with machine learning in a way I can never imagine. I had millions of records. And what I did at night is I built the technology that was to become Netramark. And it almost killed me because I was working 100 hours a week, and your brain starts feeling funny when you do that. And I had some weird skin condition come on from the stress, they said. And then I raised a couple $1 million and all my skin problems went away. I quit my job at Equifax and I built Netramark up. And then what happened was I built Netramark in parallel to what was going on at the same time. You guys are probably aware there's a lot of drug discovery companies, and artificial intelligence is quite powerful at looking at chemical libraries. And so what I did is I said, because of the perspective I gained from having such amazing training at Princess Margaret Hospital and Toronto General and Toronto Western from these psychiatrists and oncologists and computer scientists, I realized that what these pharmaceutical companies were doing and what these drug discovery AI companies were doing, where they're creating darts that you were trying to literally hit 5 different targets with. And you can't do that unless we're in a quantum world, and that's a whole other story, right? So your one dart has to hit a board, has to hit something. And what I mean by that is I already had this perspective, and this is what Netramark came. Netra is Sanskrit to see. So I built this system using this mathematics of dynamical systems and all this to infuse machine learning with this ability to see patient populations in a way that no one's ever seen before. And it was a thrill to build this, but I realized that drug discovery is a problem, clinical trials are a problem, because the actual taxonomy of disease is wrong. When someone says breast cancer, it's nonsense. It's like 17 different diseases. And Alzheimer's disease, the complexity is astounding. There's a lot of ways to get to Alzheimer's from this healthy spot that all of us hopefully are from here. And so what I did is what I decided to do is dedicate myself to helping pharmaceutical companies understand their patient populations using a whole set of tools in mathematics. Okay, so basically, let me talk to you about Netramark a bit. All right, I would have done a demo. This is kind of cool. You guys would have got a kick out of it. I'm sorry, I should have, but I'll show you some stuff. So basically, clinical trials suck. And the reason is the data from phase 2 is not predictive of phase 3. You and I can become business partners and raise $15 million and get a great phase 2. 15 million is not enough, but you know what I'm saying. And we're going to be like, we're going to be so rich. This phase 2 is going to move on to successful phase 3. No, it won't. It doesn't work because it's not predictive. And I was trying to build the technology to explain that. Why wasn't it predictive? So the core issues are basically this. I already said the first one. There's a bunch of potential adverse events because you're literally drugging people and interrupting their molecular circuitry. Drugging someone is very serious, right? Even when you're having fun on a Saturday night, but that's another story. But it's serious because you're literally altering the homeostatic systems that are keeping you healthy and alive. And the other side is placebo response. What I've learned in my journey, especially when I was a scientist at CAMH, is that I was able to induce diarrhea in 40% if I got a room of 100 people and they were all lactose intolerant, and I gave them cheese which had no lactose in it, but I said, oh, I made a mistake. I gave you cheese with lactose. 40% of them will end up, or a bit lower than 40, but still significant number of people will get sick. Placebo response is not understood, and I'm not saying that our minds have this superpower. I just know they screw up clinical trials. And so that's one of the things we really go after. I'm going to show you an example of how we were able to help a clinical trial by using artificial intelligence in a very specific way. And the other problem is that the biggest problem, forget about this, the real problem, you guys as mathematicians or as mathematical scientists are on your way to becoming that, the real issue is that patient populations are small, clinical trials are very expensive, and a phase 2 trial, I'm lucky if I have 100 people in there. And so the data sets are small, and so there's a whole group of companies now trying to get into this space by using old data that they've collected from other trials, and they want to apply it to this one, and it doesn't work because placebo response changes depending on the sites, depending on the look of the doctors, depending on attitudes of the patients, and we're biochemically different than we were before because of our foods and because every single person here has had COVID. I've had it 5 times, and my nose shut off 3 times. That's a real thing, right? I'm biochemically altered. And there's other things going on. We're constantly changing. Humans are constantly, we're not evolving, but we adapt and we're changing, and there's mutations and so forth. And the fact that these small data sets don't represent the universe. So I take 100 Alzheimer's patients or 100 schizophrenia patients, like I'm going to show you, it doesn't represent the universe. So how do you solve this problem with AI? And so the problem is this. These methods that all of you, I really recommend you become experts on, because it can give you a job. There's a lot of money in this area. But the statistics and regression, and even, by the way, you know what I learned from Equifax about neural networks and gradient boosting and random forest? You know what I learned from having millions of objects? Gradient boosting is the best thing ever. It is, if in terms of replication for prediction, gradient boosting machines, any type of ensemble tree beats everything, unless of course your data has pictures or sound or things like that, then neural networks win. But anyway, all this stuff, the problem with all of these methods is that, well, it listens too carefully to what you're telling it. Right? What I mean is, and this is something for this next generation of scientists in the maths to understand, is that these methods are great when you have an extreme amount of confidence in the dependent variables that you have. And when you're talking to doctors who are labeling patients, they have very little confidence because they don't even understand the disease themselves. They're seeing patients, they have a very hard job. They have patients coming into their clinical trials. They want to help them. They say, okay, they take a scale, they do some measurements, and then they say, this person has ovarian cancer and they were not a responder to our drug. Well, that's very complex. And that complexity gets simplified when I go to my machine learning and I say, here, dependent variables, 0 and 1. 0 the non-responders, 1 are the responders, and you're hiding all this complexity. And so what I did is I figured out mathematically that there's these creatures, these dynamical systems that have memory. They have this long-term memory. So you guys are probably excited about large language models. So what happened that allowed the large language model revolution to come is the attention mechanism, multi-headed attention. So back in 2010, when I was doing basic mathematical research for AI, I constructed a similar attention mechanism, but for feature selection, for understanding how variables relate to each other. So what I figured out was, so this is some of the math, what I figured out is, if I line up my variables and I create an artificial time, t prime, and I can just say, oh, these are events happening and some time, right? And so what I figured out was there's this creature that I can apply to large chunks, right? And what happens is this variable can be given, in some sense, some sort of heat kernel that allows them to collide with the other variables. And then through a reinforcement process, I was able to get much lower into the causality of what was separating things. So what I mean is this. So if you look over here, that red, so that picture on the top is what a very good machine learning expert was able to do on our team with lung cancer data. The red group, so this is non-small cell lung cancer, right? Awful disease, don't smoke cigarettes, okay? You shouldn't vape either, but anyway, right? So adenocarcinoma, squamous cell carcinoma. So these are subtypes of non-small cell lung cancer. And you can see the machine, it figured out these 4 variables. These are transcriptomic. I don't know if you know what that means, but we can measure how much of each protein are being made by counting copies of the mRNA from our genes. And so this is like gene expression. And so it's a very powerful tool. And you can see it separates very beautifully. Good job. That's good. Completely useless in practice commercially, because we already know there's an adenoma squamous, because this was given to us by surgeons who gave these diseases such stupid names to begin with, like non-small cell, literally from observing it during surgery. Okay, this is what we're dealing with. Literally, when you go out into the world, we're dealing with completely obtuse labels, and this is a problem. This is what Netra did. Netra created 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different classifications on its own. And it's not using principal components, and it's not using TSNE, it's not using UMAP. Those are all nice, very tools. They do the above crap. This is something else. This is literally pulling out, why are these patients behaving this way? Why are these patients surviving, et cetera? In this case, what we did is the reverse. We let NetAI talk to us, and then we went to experts, and we said, we discovered this stuff, and this is what they told us. They said, these 2 groups are interesting because of the same disease, but these people have the signature for extreme aggression in the adenocarcinoma. I'm like, oh, that's pretty cool. And that's just pretty smart. And figured out that there's other structure. And then it kept going. And so what we did is we wrote a paper that basically said, and you can look it up, if you Google my last name, Gerarci, with lung cancer, it'll pop up, right? One of the papers will pop up. But basically what we did is we basically came back and said, oh, look how great AI can be with small data sets. We discovered a bunch of stuff that we know, plus we revealed a completely new target to attack lung cancer with, a protein called PIGX. Now, PIGX has an interesting story because the referees bashed us for a year, like, oh, we never heard, you don't have any evidence. I'm like, yeah, it's just a hypothesis. Let's not take ourselves that seriously. It's math. Like, we're using math to come up with a hypothesis, and maybe one day somebody is going to actually do something in a lab. But this is what happened. August 15th, I'm in San Francisco in the audience at a brain cancer conference, because we do a lot of brain cancer work. And the first author on this paper was one of my students at U of T, actually. His surgeon, because he had brain cancer, was on stage. And I didn't know that was his brain surgeon. So I emailed her because she found something in brain cancer that was connected 1 degree from PIGX. Because I think in graph theory, right? So to me, this is all networks. So I'm thinking, oh my God, the thing that she's talking about is right next door to Big Hex. They're doing something together. So I emailed her, and she emailed me back and said, yeah, great, let's work together. By the way, I'm so happy that you got Moses, the student, to do this work. I was his surgeon. And so it was a complete fluke, and we connected around this target that we discovered. Anyway, let me tell you about the process. So now this is business. This is the way we do business, just so you guys can know like how much work, like it was years of work, and then finally we had a stream. So basically what we do is We take all their data, everything, whatever they have, genetics, proteomics, stuff from watches, personality measures, everything, because we have to deal with placebo, and I'll show you that in a sec. And then what we do is basically our company has to scale on this front because AI is not smart enough to organize and integrate data, because sometimes the data we get is a mess. Like sometimes some of the variables are in French, right? Like how are we going to get an AI smart enough to recognize, anyway, so if any of you guys want to start a company with me that does this part, I'm in, I'll invest. Okay, so this part, and this is where we have to, this is the most expensive part, because we have to hire talented people that know how to wrangle data and understand it. Okay, and then what happens is it goes into the Netra AI, which is actually quite a complex path. But there's all these different modules. There's a placebo module, there's Netra Play, which allows you to actually interact with the data sets. I'll see if I can show you some of that. And then Netra GPT, which is basically now, we have this system that literally comes down and explains, like, we discovered this subset of people, this is what's driving it. This information goes into a large language model that we've engineered, and it literally writes its own report now. It's amazing. It's astonishing. I'll show you what we've been able to do. And then what we do is we deliver the insight to the pharmaceutical company. We meet with them and we help them. And then what the pharmaceutical companies do is they look at their next trial and they say, all right, we're going to eliminate people. We're not going to include people that have these characteristics, and we're going to include people that have these characteristics before you randomize. And you're allowed to do that as long as you tell the patient way after this process is done whether they fit into that profile or not. And so that's precision medicine. That's what you guys have probably been hearing about. And that's what we're doing. So here's an example of a schizophrenia trial where we successfully did this, just to give you an example. It's a small data set, 87 people. So there's 87 people, 39 placebo, 48 in the active arm. Okay, and they came to us because they passed their phase 2. They got a p-value of a 0.04. So you understand, after they do all this, they have to look at the endpoint, whatever they're measuring, to determine if the drug is doing something. There's various ways, depending on the disease. But these people, whatever they're measuring, came out as significant, but the person running this trial is really smart. And he came to me and he says, a 0.04 is not going to be able to be, we're not going to pass our phase 3. And so what we did is we studied, we let the AI eat these patients. What we do is we organize the data in these simple tables, right? And you can have 1 million variables in there. It depends on what you're doing. In this case, the endpoint comes from something called the PANSS. It's a way of measuring schizophrenia severity and the different subtypes. And so this is what happens, okay? So what happens is you end up with, so this is the placebo group, and you end up not being able to explain everybody. For example, there's this mixed group here. So what you're looking at here mathematically is this. You actually have this function has the configuration of this iteration is basically it's basically R2. So what it does is it takes variables from Rn, and through the configuration space, it gives you some representation of R2, right? And in the representation of R2, you get collections of groups that are unexplainable, meaning that if you treat this as a hypothesis, there's no statistical power to it. And then you have other subsets which are highly explainable. Okay, and so this is an example. So here you have an example where these people are placebo non-responders, this big oval over here. Oops, wrong one, right? Oh, sorry. Right, and this group up here are placebo responders. And what we can do is we can derive what's going on with them. And what's very interesting now from a modern perspective, is the system pulls out these items, it gets put into a large language model that's been trained on huge amounts of medical literature, including psychiatry, and it can start interpreting, like we didn't know why supine respiration was caught in this. What is that? And so what happened is we actually did a test. We got doctors and scientists to interpret it, and the large language model. And the large language model said it had to do with the autonomic nervous system, being engaged in causing a placebo response. And it's what the doctor said too. And it's really cool, right? It's really cool that we're able to get this visibility from what all this math is doing. And now, because of the way we built it, it can talk to large language models. Anyway, the same thing for the drug responders. I'm not going to get into why, but basically there's a technical reason why you have this highly tight group of non-responders. And these are the variables. And what you have to start thinking about when you're doing this is, do the variables actually separate my population? And if I have too much criteria, if I use all of these things and all of these things to enrich my trial, how many people am I going to find that exactly matches these barriers? So now you have to start playing this game of like, we want the ideal patient. So the ideal patient would be someone that fits into all of these criteria, but they don't exist. So you need to have what's feasible, right? And when you do that, you actually evaluate, okay, these ones are effective, these ones are potentially effective. We're going to choose these 4 because it's actually doable. You shrink your clinical trial, but the effect size becomes huge. And this is what ends up happening. You turn your p-value from a 0.04 to a 0.002, right? Which is extremely effective. And we do this because by choosing your population, right, randomizing, and then going forward, the FDA will look at what you're doing and say, okay, you're following a randomization scheme. You're selecting patients for a certain thing, so we have to put that on your drug as a label. This is a biomarker. This is the precision medicine part. And then, but they end up passing their trial. So you give up the ability to treat everybody with your drug, which, trust me, in the beginning was a hard sell, because people didn't hear about this. They're like, no, we want our drug to treat everybody. But that's why you have so much ineffectiveness and illness being caused by your drugs. This is a way to make it much safer and to actually treat people who need it and who will get a result. And this is actually a very cool thing. We actually won a global competition using this AI. I'll just tell you this and I'll finish up, is what we have here is, this is ALS. ALS is a horrible, rare disease that basically is what Stephen Hawking had, right? I'm sure everybody here knows Stephen Hawking. And the competition got this public data set, transcriptomic, that I helped process. And then what we managed to do with this paradigm was we were able to, so let me explain something to you. All of these blue people that you see at the top, these are people for which the disease starts in their limbs. So they start having trouble walking, right? And their fingers stop working, and then it progresses. That red little dot up in the top is actually people that have it start in their throat. They have trouble swallowing, right? It's a really horrible disease. And what we were able to do is the machine pulled out this map of the patients, and it pulled out a bunch of variables, which is fascinating, and I'll tell you why. And we were able to literally, in an interactive way, zoom in and pull this group of people out. And what was amazing, it's just a small group of people, but statistically significant, because what was driving this subgroup, this vulnerability, was something we call axonal transports, these proteins that carry other proteins across your axons. And your motor neurons are extremely long. So these things have to work harder to carry these proteins down your neurons. So the rest of their body that are not motor neurons are not affected by this genetic malfunction, but these very long motor neurons get messed up. And the cool thing is, when it explained itself, it's exactly the cutting edge of what's going on in ALS today. And I was able to present this on a panel in Boston just this year, right? So what it means, what I'm trying to show you here is that, so the punchline to all this is that AI by itself is great, but it requires mathematics to take us to these next levels, and that's what's happening. I actually have a big belief that people like you can literally change the way we engage with medicine and other industries, because a lot of people out there are just convinced, oh, they're built, I'll just use a deep neural network. But there's so much more that's possible. And your training in the mathematics and mathematics in general, it can be really used to bolster the next generation of tools, like the way I did. And it's been a great journey, and So now what's happened is I sold this company. I bought a nice car that my wife has right now. And I have the crappy one. And I moved up north and I reinvested because I know where clinical trials are going. And so we're public now and we're pushing. And there might be opportunities in Netramark for you guys in the future. But the other thing you should think about is that the way the world is going, there's a huge need for people with your kind of minds to take this in the right direction and to also take it in a safe direction. And that's a whole other topic.